Zinc fingers are historically the most amazingly programmable approach to getting DNA-protein interactions. So if you want to take a protein to a particular place on DNA uniquely, it's the way to go.
The other interesting thing about the proteins is that you can fuse functional domains to them. So you can fuse things like nucleases, transcriptional regulatory domains, and then you can use the zinc fingers to direct those functional domains to specific genomic locations and do things like modify specific gene sequences or modify expression of neighboring or nearby genes. The other appeal of them is that they work in a multitude of different organisms. They've been shown to work in everything from yeast all the way up to human cells.
We started working on zinc fingers in the late 80s when we were looking for ways of applying double-stranded DNA on chips where you could synthesize a forest of different DNA molecules and find out where the zinc fingers would bind and try to crack the code between the zinc fingers and the DNA. At that time, the code was not nearly as evident as it is today. And then today we're interested in them as ways of getting homologous recombination to work very efficiently in mammalian cells, plants, and bacteria.
In terms of discoveries that we've made, I think perhaps one of the more significant ones was the development of the so-called open platform, so the oligomerized pool engineering platform. And really what that did was provide a rapid method that was also robust and could be practiced utilizing publicly available methods to be able to engineer zinc finger proteins. And we published that method about 3 years ago and have made those reagents available to academics who are interested in practicing the method. And I think it's been adopted by, certainly the reagents have been requested by a large number of groups, and we believe that many of them have been practicing and making their own proteins using this method. So the code really, I think, stems from some early work, structural work, that suggested that there might be a simple one-to-one relationship between amino acids, specific amino acids within the zinc finger, and then bases bound by that zinc finger.
What is the zinc finger code? It's basically a way of going from any target DNA that you'd like to bind to double-stranded DNA to the protein sequence that would bind to it. Or in principle, given a protein sequence in a zinc finger, you could figure out what DNA it binds to.
And I think that in the early days, the hope is that if one could elucidate this code, then one could simply use it to reprogram or redesign zinc fingers with novel DNA binding specificities. Like most things in nature, it turns out that the code is somewhat useful, but is not the entire story. And that if you really want to get zinc fingers with optimal DNA binding, you have to account for other effects. So for example, zinc fingers exhibit what we call context-dependent effects. That is, the activity of any given zinc finger is going to be influenced by the identity and by the sequences of its neighboring zinc fingers. And so because of that, the one-to-one correspondence doesn't always work as well as people had originally hoped. And so accounting for this context dependence means that you then have to look at more combinatorial possibilities, which in turn makes it more challenging to engineer the zinc fingers. So the code, I think, is a useful starting point, certainly for design, but the actual activities and binding of the zinc fingers did not turn out to be as easy as I think those in the field had hoped.
Well, probably the most cutting edge would be gene therapy. And gene therapy had a setback about a decade ago when viruses were used to kind of randomly integrate in the genome, and that this provides an alternative where you're doing very precise integration, not just in a safe haven where it's safe to put something here, but where you're changing the part of the genome that you want to change. So the most cutting edge application then would be changing the human genome for gene therapy. Ideally taking your own cells and changing them and putting them back.
That you can take cells from a patient with a genetic disorder, for example, and remove them from the patient and then modify them outside the body. So what we refer to as ex vivo modification of the cells. And basically the approach would be if there's a gene and a mutation in that gene that's responsible for a disease, one can contemplate using technologies like zinc finger nucleases or perhaps talons to then correct that mutation and turn it back into a wild-type sequence. And then taking either those cells directly or potentially modifying their differentiation state and then transplanting them back into the patient. And I think the exciting possibility there is there are at least two exciting possibilities that are sort of different from other things that have been done in the past therapeutically. And one is that you're actually correcting the gene itself instead of trying to replace it with a wild-type copy. The other exciting thing is that you're putting in patient cells that come from the patient themselves, and so therefore you may be able to avoid doing things like immunosuppression and complications that are associated typically with transplant, like things like graft versus host disease.
The most cutting-edge application that I have affection for is deleting the CCR5 gene, which is one thing that Sangamo has in phase 1 clinical trials, inspired by a cell therapy, hematopoietic stem cell transplant about 4 years ago, where you make the patient resistant to HIV because CCR5 is the HIV receptor. And that's very clever and looks very promising.
So I think that's the excitement really behind zinc fingers, is that one can make customized versions of DNA binding domains and then direct specific functional domains to particular location within a genome.
What technology made zinc finger research possible? Well, certainly crystallography was really an important prerequisite because it told you the angle and the sort of amino acids that were making contacts with the sort of base pairs. So that helped to do rational design. But it turns out the zinc fingers aren't quite as rational as they look in an X-ray crystallographic diagram. And so you need to do a little bit of exploration and selection. So that the bacterial 2 hybrid is an important technology for searching through and refining the DNA-protein interactions. Most of the going from crystallography to bacterial 2 hybrids to screening through has been relatively low throughput. We now are reaching an era where literally we can have a computer design as many synthetic genes as we want. From a chip you can make thousands of genes. You can make tens of thousands to millions of oligonucleotides per chip. And so that's greatly enabling. The computer-aided design software with improved rules should be a big advantage. Being able to get these things into mammalian human pluripotent stem cells will be an important technological combination to expedite this, because some of these things will bind well in vitro, but not necessarily in the cell that you want. And the cell you want is something that can turn into almost any other cell. So the pluripotent stem cells would be an important technology for the future.